Hi, Dr. Woeller here for Integrative Medicine Academy. I want to welcome everybody to this presentation. The title of this lecture is Mitochondria Melatonin Production and its role in reversing aerobic glycolysis, and specifically implications for inflammatory-induced diseases, and one of those would certainly be cancer. And I thought I'd do this particular talk because it ties in very much with a lot of the research that I've been doing lately with regards to mitochondrial issues, and certainly what we're hearing a lot about now with chronic illness being linked directly to mitochondrial dysfunction, and in so many ways, whether it's just from internal factors, genetic issues, mycotoxins, chemicals, etc. So I thought this would be an interesting lecture to put together, and specifically looking at melatonin. One of the things I didn't realize was that the role that melatonin actually plays in mitochondrial function, because I think a lot of us assume that it's only the pineal gland that produces melatonin, and that's far from the reality. So for those of you who have not heard me speak, I've been an integrative and functional medicine physician since the late 90s. I do a tremendous amount of speaking online as well as in person. I've written a number of books. I really love clinical education. I'm also a practicing clinician. I've been the clinical educator for Great Plains Laboratory for many years. I actually put together their organic acid test seminar, as well as education director for our Integrative Medicine Academy. I'm also the co-founder of this academy. I'm also the co-founder and education director for a website called Functional Medicine Clinical Rounds, which is a membership site for healthcare practitioners who are seeking assistance in lab interpretation and case presentation, clinical troubleshooting, etc. I'll talk more about some of these a little bit later. And then in my own practice, I've been working with the autism community for many years, as well as patients with chronic and environmental induced health problems, whether that's autoimmune, GI issues, mold, etc. So some of this material actually comes from a course we have through Integrative Medicine Academy called Mitochondria Mastery Course. You can go to mitochondriamasterycourse.com to get more information. This is a course that's coming up, and really excited about this course because there's so much information to go through some of what you're going to hear today. So when we think about mitochondria, so here's some questions that I asked myself when I was putting this together. And this is specifically looking at mitochondria function and its relationship to melatonin. So essentially, what is melatonin and what's the difference between pineal production of melatonin and mitochondrial melatonin? What are the mitochondria, or mitochondrion essentially, which would be an individual mitochondria. Mitochondria would be the plural aspect or plural form. And then what common metabolic pathways exist in the cells for energy production? We'll talk about what's called aerobic glycolysis, and then some additional information and considerations regarding melatonin. So first off, let's just think of melatonin pineal versus mitochondria. So some basic information. The melatonin it's often thought of as just a hormone produced by the pineal gland. And we know that it's a chemical compound involved in the sleep-wake cycle. So if you see over here on the right, we can see the conversion steps to melatonin. So tryptophan becomes 5-hydroxytryptophan, becomes 5-hydroxytryptamine, and acetyl serotonin, and then melatonin. And I'll talk about those again shortly when we talk about or look at the sequence of the biochemistry within the mitochondria. So again, melatonin, its role in the sleep-wake cycle is fundamental. We know that this is extremely important for our overall health, and just to kind of help reset normal biological function within the brain and the nervous system, etc. So we all can feel the effects when we don't have enough melatonin or don't get a good night's sleep. Essentially, we're just we don't function very well consistently if that goes on for a long period of time. So melatonin has this rhythm effect. So it's obvious, right, from a pineal perspective, melatonin levels generally peak in the middle of the night, or they should be, and then the levels will drop off quickly in the morning when we're exposed to sunlight. But notice in this particular graph that the melatonin levels don't actually fall to zero. So there is some melatonin produced throughout the day, and then it starts to ramp up again, particularly in the evening time after sunset. So a lot of times melatonin is thought of as like the molecular equivalent to darkness, for example. And so light, right, triggering through the eyes, along the retina of the eyes, there's a connection to what's called the suprachiasmatic nucleus. That input from light, whether it's the sun, whether it's light in your home, whether it's blue light coming off the TV or your computer screen, or your iPhone or your tablet, okay, it will communicate through these nerve pathways and through the suprachiasmatic nucleus. And if there's enough of that activation, it will actually inhibit the pineal gland from producing melatonin. Okay, so, and then the reverse obviously occurs when we're in darkness. We should get more melatonin production. And what's interesting about melatonin, just from a pineal perspective, we know it is involved in that sleep-wake clock, that circadian rhythm, that cycle. And that also has communication into the hypothalamus, which then has influence along with other inputs throughout the body, particularly the adrenal glands, the liver, etc. The other thing to keep in mind is that all of us, as we age, will lose some of that natural melatonin production that occurs within the pineal gland. So in general, in a healthy individual, we're getting like peak output of melatonin, usually between the ages of 10 to 20, and then it starts to drop off. So by the time we're in our 40s, our melatonin is, continuing to decline. And then as people move into the older years, 60 and above, is where things really start to significantly drop from a melatonin perspective. So a recap, pineal melatonin sets that circadian rhythm. It is affected by dark and light exposure. It is rhythmic. And what's interesting is that the pineal melatonin will have an effect on blood levels. That particular type of melatonin that's in the blood can actually be transported into our cells. And so it may be coming from the pineal gland, but it does have an influence on cellular activity throughout our body because it can be transported in the blood. So there is a mechanism where we can get some of that melatonin into our cells and may even influence mitochondrial levels. But what's really interesting is that only 5% or less of our total melatonin production in our body comes from our pineal gland, which means about 95% of melatonin is actually produced in our cells, specifically our mitochondria. And the cellular melatonin doesn't really contribute to any significant degree of circulating melatonin in the blood. So pineal melatonin will contribute to circulation throughout the body and the blood, but mitochondrial produced melatonin does not. Now, the mitochondrial melatonin can have a local effect, what's called a paracrine or an autocrine effect. So a paracrine effect is it may help influence activity of an adjacent cell, wherever that is in the body, or that melatonin that gets produced from that cell can sort of have a feedback mechanism within that cell, essentially. But it's not contributing significantly to anything in the blood. So we know that melatonin's major role, other than affecting the circadian rhythm for sleep, is it's acting as a potent antioxidant within the brain, the nervous system, and essentially all cells that contain mitochondria in the body. And that's really its primary role, or one of the primary roles. And there's a lot of research. So this is actually an article that came out in Life Sciences 2004, Melatonin and Mitochondrial Function. Okay, they know now, or we know now, that common features in various diseases like Parkinson's disease, Alzheimer's disease, epilepsy, sepsis, for example, there is the existence of mitochondrial damage that can occur because of ongoing oxidative stress. And that melatonin in these particular circumstances acts directly against these toxic oxygen and nitrogen reactions or compounds that get generated in the cell that is putting stress on the internal mechanisms of the cell, whether it's the mitochondria or the endoplasmic reticulum, for example. We need melatonin to help buffer against these oxidative reactions to support proper functioning of the electron transport chain that's sitting within the intermitochondrial membrane. So another paper, 2011, melatonin mitochondrial interplay in health and disease. So in quotes, melatonin is a known powerful antioxidant and anti-inflammatory, and increasing experimental and clinical evidence shows that it has beneficial effects against these oxidative and nitrogen-based reactions that lead to mitochondrial dysfunction. If we hold that thought, we can see how melatonin from a supplement standpoint could be beneficial for people who are compromised because of mitochondrial dysfunction, regardless of what's causing the mitochondrial dysfunction. So let's look a little bit more at the mitochondria, what's often considered to be the powerhouse of the cell, and go through this a little bit more in depth. Okay, so mitochondria, we know that these are the energy factories of our cells, right? They're producing the adenosine triphosphate that's needed as a critical chemical throughout the cell, but also the entire organism for metabolic and physiological action. And the amount of ATP that's needed daily for just normal basic function is huge. It's almost incalculable how much is needed on just a daily basis to maintain normal function. If you think about, I'm sitting here giving this presentation, you're sitting listening to that. Well, that requires activity at a cellular level through your skeletal, musculoskeletal system, your cardiovascular system, your nervous system, etc. And so when problems actually occur in the mitochondria from intrinsic biochemical dysregulation or outside influences like toxins, for example, we know that these can lead to health problems. And there's a lot, right? This is just a short list. Mitochondrial dysfunction and associated diseases. So neurodegenerative diseases, cardiovascular problems, diabetes, metabolic, autoimmune. See a lot of mitochondrial issues in the autism community, as well as gastrointestinal chronic fatigue, and then cancer. So the list goes on and on if you really start to branch this out and look at different types of disease states. And so fundamental to the cell is the mitochondria, which contains two membranes. We've got the outer membrane and we've got the inner membrane. And in between the inner and outer is that intermembrane space. Sitting within the matrix of the mitochondria is what's called the Krebs cycle, also called the citric acid cycle. And then embedded in the inner mitochondrial membrane is what's called the electron transport chain. So as we move substrate through the Krebs cycle, there's two connection points that occur. We've got complex 1 and complex 2. Complex 2 is activated through or is connected to an enzyme called succinic dehydrogenase. Nicotinamide adenine dinucleotide hydrogen is actually produced in part in glycolysis as well as the citric acid cycle that connects into complex 1. We actually get maximal ATP production when we engage the electron transport chain at complex 1. We can block complex 1 or complex 2 and still get some ATP production, but it's not as robust. The main point that leads to cellular death and death to us is when you completely block complex 4. So cyanide and carbon monoxide essentially are very toxic substances that affect the mitochondria, and inhibition of complex 4 can be lethal. So mitochondrial dysfunction can occur from many different reasons. Ultimately, a depletion or a decrease in ATP production is sort of the end result. And so whether it's a loss of electrical potential of the inner mitochondrial membrane, or we disrupt those different complexes that are part of the electron transport chain, or just block the ability to get substrate into the mitochondria, all of that can lead to problems in the mitochondria, leading to mitochondrial dysfunction. A very interesting article I would encourage you to read, you can pull this up online, called Mitochondria Fundamentals to Life and Health. This is an article that came out a number of years ago by Dr. Joseph Pizzorno in Integrative Medicine. And so these bullet points actually come from this article. So think about this for a sec. So the amount of ATP produced daily from a healthy individual at rest is equal to their body weight. So think about what that means of how much ATP we have to produce when we're active. With maximal activity, the amount of ATP can increase to 1 kilogram per minute. So just a huge increased demand. The other key thing is ATP is not stored in our cells. So we have to produce it moment to moment, every second of our lives, for us to be able to function normally. In fact, the average cell, about 25% of the volume of that cell is occupied by mitochondria. And I've actually seen some articles discuss like heart cells, for example. I'm not saying all of them, could even be upwards of 40%, the volume occupied by mitochondria. And so each cell contains thousands of mitochondria. The more metabolically active, the more mitochondria exist in that particular cell. Okay, and so an average cell, for example, could produce upwards of 10 billion ATP per day, for example. So just a huge amount metabolically that we need just to function normally. So let's look at some of the basics of cell metabolism, kind of typical metabolic pathways. We're not going to go through all of them. We'll do an overview. So one of the key things to look at on this particular slide is, first off, pyruvic acid, and then the second is acetyl coenzyme A. So notice that proteins, carbohydrates, and fats can all funnel down to acetyl coenzyme A. Now, acetyl coenzyme A can also go off and form what's called lipogenesis to reform fats, for example. But from the acetyl coenzyme A position, we move into the citric acid cycle, into what's called oxidative phosphorylation as part of the electron transport chain to produce huge amounts of ATP, and essentially between 36 to 38 ATP, if we're just even talking about glucose, for example. We can also form lactic acid, okay, or we can reshuffle things and form new glucose essentially through gluconeogenesis. But getting down to this level is fundamental and extremely important for cellular metabolism to get the biggest bang for our buck metabolically from ATP production. So here's just another view of glycolysis. So here we have a molecule of glucose. We input some ATP at a couple points strategically. And at the end of glycolysis, we form pyruvic acid. So for one molecule of glucose, we form 2 molecules of pyruvic acid. So that essentially is glucose metabolism occurring within the cytosolic fluid of the cell. This is just another viewpoint, just a different angle. So here we have glucose that ultimately gets converted into 2 molecules of pyruvic acid. So we have to expend some ATP up front and then we gain some ATP on the back end. So by the time we get to pyruvic acid, we're at a 2 net ATP because we had a spend 2 up top. We broke even about midway through, and then we gained ATP at the pyruvic acid level. And so essentially this is what's called anaerobic glycolysis because it doesn't require oxygen. So it's an efficient but not highly productive form, meaning we're not getting huge amounts of ATP from this, but it does serve a purpose, particularly for immediate ATP need. But the key step becomes when we start to move into what's called aerobic metabolism, where we're now incorporating oxygen into the equation, is to get pyruvic acid into the citric acid cycle. Okay, so that is a key, and there's an enzyme that makes that happen called pyruvate dehydrogenase complex. I'll come back to that here shortly. So just a very simplistic way of looking at this is glucose becomes pyruvate through anaerobic glycolysis. Pyruvate, we get some ATP from that, by the way. It's about 2 ATP, for example. And then pyruvate can enter the Krebs cycle, get converted to acetyl coenzyme A, which then gets spun through the Krebs cycle, substrate pulled over into the electron transport chain to produce very large amounts of ATP, 36, 38 ATP, for example. And so that becomes aerobic respiration. So aerobic meaning we need oxygen present in order for that to happen. So coming back to this view again, we've got our outer membrane, we've got our inner membrane. Inside the matrix, we have the Krebs cycle. We've got the electron transport chain that's embedded in the inner mitochondrial membrane. We get most bang for our buck metabolically when we can engage metabolism at the mitochondrial level through the electron transport chain, because this is where we're going to get the most of our ATP produced. So the electron transport chain is an interesting area to study, and we get into this very great detail in the Mitochondria Mastery Course because there's a lot of things that feed into it. So you can see here we've got CoQ10, for example, that is utilized in electron transportation within the various complexes. We've got NAD, for example, that initiates activity at complex 2. We've got succinic dehydrogenase linked to riboflavin, for example, which engages complex 2. I think I meant to say NADH at complex 1, riboflavin at complex 2. But ultimately, at the end is our ATP. What's interesting is as we're moving electrons, okay, from one complex to the next, as this moves down this chain reaction, so electron transport chain, we're also getting an outward movement of hydrogen ion or a proton that carries a plus charge. And when we get enough of those plus charges that are accumulating over top what's called our ATP synthase, sometimes called complex 5, we get an inward movement of those protons through an electrical chemical gradient that causes, or there's a rotational effect on these oval-shaped proteins that brings together adenosine diphosphate and inorganic phosphate to make our ATP. So as we get one rotation, we pop off one of these ATP, and then that goes essentially to the accumulated pool of ATP at the cell. I won't get into it here, but the ATP actually that gets produced through ATP synthase activity is stabilized by magnesium. So magnesium is a very important mineral in the mitochondria to help stabilize the ATP against further reactivity. Oh, I wanted to mention one thing. Right here, strategically, here's the inner mitochondrial membrane. And sitting on the outer edge of this inner mitochondrial membrane is cytochrome C. And it sits on that outer mitochondrial membrane for a reason. It's a small heme protein that's loosely bound to the outer surface of the intermitochondrial membrane, and it helps to shuttle electrons from complex 3 to complex 4. When the mitochondria is stressed, or the mitochondria is being attacked, let's say from a mycotoxin, for example, or something that damages that intermitochondrial membrane, the cytochrome C can get released. So it actually, let me just go back. This thing wants to. So it can release from the intermitochondrial membrane, and it can move into the cytosolic fluid of the cell, okay, and trigger what's called apoptosis. So it basically interacts with different enzymes that are sitting in the cytosolic fluid of the cell as a signaling mechanism to say, hey, we need to eliminate ourselves. We need to sort of, basically what the cell does, or the mitochondria does, is triggers a reaction for that cell to commit suicide. So it removes itself from its ability to create any potential harm down the road. So unfortunately, that can have a runaway effect where we start to lose mitochondria too. So that it's an intrinsic mechanism that's inherent to our body, which is a good thing, but too much of it can be problematic, obviously. And mycotoxins, for example, are a major trigger for damage of the intermitochondrial membrane and the release of cytochrome C. So we'll just bring it back to the level of melatonin for a second, just as a recap. So melatonin's major role, other than affecting the circadian rhythm for sleep, is as a potent antioxidant at the cellular level. So we talked about anaerobic glycolysis. We talked about oxidative phosphorylation, for example. Well, let's talk now about aerobic glycolysis, what's called the Warborg effect. If I'm pronouncing his name right, Warborg. So Otto Heinrich Warburg. So Dr. Warburg received a Nobel Prize in the 1930s, so 1931, for example, for the discovery that unlike other cells in the human body, cancer cells, in essentially their terminology at that time, don't breathe oxygen. So cancer cells are anaerobic mostly, which means they derive their energy without needing oxygen. So why do they do this, and how do they do this, for example? So from an oncology standpoint, this has been well studied going all the way back to when Dr. Warburg, found this effect. It's called aerobic glycolysis. And so essentially it's the observation that most cancer cells produce energy through a less efficient or aerobic glycolysis effect. So they uptake glucose, they go through glycolysis, they produce lactic acid fermentation through fermentation reactions. And they do this in a cell despite the fact that oxygen is present, meaning it bypasses the citric acid cycle or the Krebs cycle, as well as oxidative phosphorylation of the electron transport chain. And it's suspected that other inflammatory conditions may have the same effect. Okay, so it's suspected, for example, that aerobic glycolysis or the Warburg effect may play a role in other inflammatory disorders. And it's a less efficient way for our body to produce ATP, but from a cancer standpoint, it actually is efficient because it wants rapid fuel, for example. And there's a lot of other downstream effects that happen at the mitochondrial level that can sort of buffer against cancer development or just runaway, cellular activity involved in inflammation. But the bottom line here for just this presentation is understand that this is an effect that occurs. It's almost like the cell gets hijacked, essentially. But the aerobic glycolysis can actually be a normal mechanism in certain cells, in certain circumstances, for a specific purpose. So for example, within the immune system, and this specifically is looking at adaptive immunity, certain T cells, when activated against an infection, will mobilize this aerobic glycolysis effect for immediate ATP utilization for an immediate immune response. So there's an aspect to this that's actually normal within some of our cells, and specifically our immune system. So this is a fascinating article. And my recommendation, if you're going to pull one article from this presentation, I would recommend this particular one called Melatonin and Mitochondria Mitigating Clear and Present Dangers. It comes from a Dr. Russell Ryder and others. And he's been a long time melatonin researcher, particularly mitochondrial melatonin. So let's go through this. I'm going to go through this particular slide through a series of slides here as we sort of build on this information. Okay, so glucose gets transported into the cells through our GLUT transporter mechanism. And then that glucose will go through glycolysis, as we just discussed, to form pyruvate. Now, pyruvate will move. This is in normal oxidative phosphorylation reactions. So pyruvate will move into the mitochondria. So now we're inside the matrix of the mitochondria. And pyruvate will get converted to acetyl coenzyme A through the actions of an enzyme called pyruvate dehydrogenase complex. And I've done lectures on pyruvate dehydrogenase complex before. One of the critical nutrients for pyruvate dehydrogenase activity is thiamine, for example. Now, notice that acetyl coenzyme A, okay, can enter the citric acid cycle. So we've already gone through that, and then that segues over to oxidative phosphorylation. Okay, so we get the major effect at the electron transport chain level for ATP production. Notice also that acetyl coenzyme A is used as one of the main cofactors for the conversion of 5-hydroxytryptamine to get converted to NAS. NAS is N-acetylcysteine. MEL is melatonin. Okay, I'll come back to that shortly. Oh, by the way, one other thing to point out. Right here is something called PDK, pyruvate dehydrogenase kinase. There is a relationship between pyruvate dehydrogenase complex and pyruvate dehydrogenase kinase. Notice that the kinase puts the brakes on the pyruvate dehydrogenase complex. So it gets really in depth when you start looking at this from a biochemical standpoint, but there's a lot of what they call metabolic flexibility in the system. It makes sense when you dive into this a little bit deeper. It's just important to understand right now that the ability to convert pyruvic acid into acetyl coenzyme A to initiate activity within the Krebs cycle, we have to have our pyruvate dehydrogenase active. And so it can fluctuate back and forth between an active and inactive state, depending on the actions of pyruvate dehydrogenase kinase. So you could think of the kinase pyruvate dehydrogenase as a brake on pyruvate dehydrogenase complex. And there's many things that initiate the brake or release the brake. So if we have plenty of ATP in our cells, or we've got plenty of NADH, or we have plenty of acetyl coenzyme A, those can activate the pyruvate dehydrogenase kinase to essentially break or slow down the actions of the pyruvate dehydrogenase complex. Pyruvate itself, or calcium, essentially inhibits the kinase. So we actually get increased activity of the pyruvate dehydrogenase complex activities, which would make sense. Basically what's saying up here is that if we already have enough energy substrates, we already have enough ATP at the mitochondrial level that's being produced, we don't necessarily need to keep shoving more pyruvate down into the citric acid cycle. So there's a brake and an accelerator that occurs in this particular system moment to moment, okay, every single day of our lives. So the pyruvate dehydrogenase complex, which converts pyruvate to acetyl coenzyme A, is regulated by a number of things. It's regulated by acetyl coenzyme A, it's regulated by ATP, it's regulated by NADH, for example. And so again, if we have enough energy in the system, that reaction will slow down. Things that activate pyruvate dehydrogenase kinase, which is the main brake on the pyruvate dehydrogenase complex, okay, right here, if we have something that activates that reaction excessively, then we're going to get a major shutdown in the ability to convert pyruvic acid into acetyl coenzyme A, and then greatly compromise ATP production downstream through the mitochondria. Well, it turns out that cancer cells upregulate pyruvate dehydrogenase kinase. Okay, so this is one of the things that happens in many tumor cells is they increase the actions of the kinase, which causes a shutdown in the dehydrogenase complex, which blocks our ability to get pyruvate into acetyl coenzyme A, which then blocks the ability to make massive amounts of ATP at the mitochondrial level, despite the fact that there's plenty of oxygen available. So the Warborg effect essentially is shunting normal mitochondrial activity towards aerobic glycolysis, meaning aerobic oxygen is present, but we're basically depending on ATP production just because of glycolysis. So remember, glycolysis gets converted into pyruvic acid. Pyruvic acid moves into the mitochondria to make acetyl coenzyme A. But in a Warborg effect, we're actually increasing the actions of an enzyme called lactate dehydrogenase A. Increases of the LDHA, okay, this enzyme here, will shuttle or shunt pyruvic acid into forming lactic acid. Now, lactic acid can change the acidic environment of the cell. You can get into a whole number of reasons of why that would be advantageous for cancer cells, for example. But the bottom line with regards to this discussion is that this Warborg effect is essentially inhibiting this, or one of the effects is inhibiting this reaction right here. Okay, so and there's a number of things that can do that. So something called HIF or HIF1 Alpha, which is hypoxia inducible factor 1 alpha. That is a activator of PDK activity. Okay, other transcription factors as well. Where it gets very interesting is where you start looking at melatonin. So notice that melatonin in the mitochondria, as we already mentioned, supports the function of the four complexes, complex 1, 2, 3, and 4. So it's helping very much against runaway oxidative reactions and free radical damage. Melatonin, we know from the blood, right? So remember, pineal melatonin can get taken into the bloodstream and can be transported into the cells. Melatonin produced in the mitochondria does not have much of an influence on blood melatonin. So we can theoretically get some melatonin produced from our pineal that could benefit, what's more than theory, it actually is known, would benefit our cellular melatonin pool. Melatonin within the cell in general will decrease cell proliferation, decrease angiogenesis, for example. Okay, it'll increase cell survival. It will decrease metastasis. Melatonin being produced in the cell or in the mitochondria helps to stabilize the electron transport chain. It helps to maximize the function of the oxidative phosphorylation effect for ATP production. The other thing melatonin does is it activates something called SIRT3 which activates superoxide dismutase, which helps to blunt or block oxidative reactions as well. Let's focus now within the mitochondria. So again, here's our pyruvate. Pyruvate gets converted to acetyl coenzyme A through the actions of pyruvate dehydrogenase complex. If we have high activity of pyruvate dehydrogenase kinase, that can block that reaction. Notice that 5-HT, so 5-hydroxytryptamine, which is serotonin, gets converted into N-acetylserotonin through the actions of an enzyme called AANAT, which stands for aryl alkylamine N-acetyltransferase. So it's basically transferring an acetyl group from acetyl coenzyme A to form N-acetylserotonin. And then that is one step away from mitochondrial melatonin, which then has the effect of enhancing electron transport chain activity. It will inhibit pyruvate dehydrogenase kinase activity. It will positively stimulate SIRT3, which positively stimulates superoxide dismutase, which positively eliminates or decreases oxidative reactions. So it is a critical antioxidant regulatory chemical within the mitochondria. This is another view from this article. So here we have 5-hydroxytryptamine. So just figure that is our serotonin, gets converted into N-acetylserotonin. That gets converted into melatonin. So it scavenges reactive oxygen species. So it basically helps to balance out runaway oxidation. Okay, it activates SIRT3. Okay, which activates superoxide dismutase, again, helping to balance against oxidative stress, and it improves the oxidation phosphorylation reactions of the mitochondria. If we don't have good SIRT3 activity, we need SIRT3 to activate superoxide dismutase. If we don't activate superoxide dismutase, we get runaway oxidative stress. So if we come back and just take another view of this slide. So again, the Warborg effect, and the discussion of this gets very in depth, more than we're going to get into here. We'll get into certain aspects of this in the mitochondria course, but researchers still haven't truly figured out all of the influences of the Warborg effect, for example, like what triggers it, what are some of the mechanisms the other things that it plays a role in. So the research in it is actually an ongoing process. It's very interesting. But the end result is known. Okay, and that is it increases aerobic glycolysis and essentially shuts down mitochondrial production of ATP. And so basically what we can do from a therapeutic standpoint is try to maintain as best as possible melatonin production and at least just understand the role of melatonin beyond just sleep. Oh, let me go back, sorry. Let me focus right here, okay? So hypoxia inducible factor 1 alpha. Now notice that when we have a lot of reactive oxygen species that are being generated in the body, one of the things that we can get is an increase in this HIF1 alpha. The HIF1 alpha will activate like cell proliferation, for example, or angiogenesis or metastasis. And from a cancer standpoint, it's not always going to do that in everybody, but it could also create problems of just runaway inflammation as well. Okay, so that's one particular role. And we know that melatonin, okay, comes over and can inhibit overexpression of hypoxia inducible factor 1 alpha. And this brings us to the second article I would strongly recommend you read. It's not a really long article. It's like 2 or 3 pages. And this came out in 2020 in Medicine and Drug Discovery, called Melatonin Inhibits COVID-19 Induced Cytokine Storm by Reversing Aerobic Glycolysis in Immune Cells. Now notice in this particular paper, they're talking about macrophages. One of the things I'd mentioned earlier from one of the early slides was that T cells can utilize aerobic glycolysis as well. So macrophages as part of our innate immune system, T cells as part of our adaptive immune system. So there is a normal aspect of aerobic glycolysis that occurs normally within our immune system. Okay, so a healthy macrophage, for example, if we go through the exact process, glucose gets transported into the cell, it gets converted into pyruvate. Pyruvate enters the mitochondria. The actions of pyruvate dehydrogenase complex produce acetyl coenzyme A that enters the Krebs cycle to increase ATP production. It also converts serotonin into melatonin, and then all of the beneficial effects of melatonin can happen within a macrophage, for example, as it can throughout the rest of our cells. In COVID-19, what they looked at was what they were specifically looking at in this particular paper is that in a runaway situation of infection, because of increased levels of pro-inflammatory immune chemicals, we shunt away from oxidative phosphorylation into aerobic glycolysis. And one of the things that induces that is HIF1 alpha. We know that HIF1 alpha is a major stimulator of pyruvate dehydrogenase kinase activity, which inhibits pyruvate dehydrogenase complex activity. And so essentially, if you look at some of the protocols through the FLCCC group, for I think criticalcarealliance.com, they actually recommend and have for a long time now the use of melatonin in the early stages or even late stages of COVID and post-COVID as well. And so it's quite possible, if we just think of this from a mechanistic standpoint, that this type of reaction could be occurring because of other infections and other inflammatory types of states, not just COVID, but other types of viral infections, other types of infections that are generating a tremendous amount of cytokine activity. So as a quick recap, the mitochondrial melatonin. So we know that it scavenges reactive oxygen species and assists in what's called cellular redox homeostasis. So basically it helps to bring things back to balance at the cellular level. It increases SIRT3 activation, which increases superoxide dismutase. It increases oxidative phosphorylation potential along the electron transport chain. It inhibits the pyruvate dehydrogenase kinase, which then allows for an increase in pyruvate dehydrogenase complex to convert pyruvic acid into acetyl coenzyme A to initiate activity at the Krebs cycle level in the mitochondria. Otherwise, you're going to have all that pyruvate that is then readily available to just get converted to lactic acid. Mitochondria can inhibit the HIF1 alpha and other transcription factors, for example, that can have a negative effect or an influencing gene activity effect within the nucleus of the cell. When it comes to supplemental dosing, okay, this is where it gets a little bit tricky because there is no set dosage specific for age or sex when it comes to melatonin. So we have to think about what we're doing as healthcare practitioners clinically. Why are we using certain things and for what type of patient are we using them for? Melatonin supplementation has no established what's called LD50. So LD50 is the lethal dose that would cause death in 50% of animals given that particular substance. From my research, there's no established toxicity dose linked to melatonin, at least that's known in current human and animal studies that I have come across, that I've heard about, for example, or what I've heard other authors speak of. There's no established duration of time set for melatonin use. So the duration of use is really based on why it's being used. So again, for most of us and most people, they're taking melatonin to assist with sleep. And we can now start to appreciate even more why proper healthy sleep patterns are so important for overall health, because some of that pineal production of melatonin has an influence on cellular activity at the mitochondrial level. So the reality is, you'll have patients that will ask, well, how long do I use melatonin for? Or what's the dose? Or my child's 5 years old, how much do they use? And the reality is melatonin in people with chronic illness, the length of time in using it is really open-ended. There is no, that I know of, a set time to use melatonin, particularly people, whether it's in cancer therapy or chronic illnesses of various forms. The other important aspect of melatonin supplementation is that it won't turn off natural melatonin production. There's no known feedback loop within this system. So conservative dosing amounts are typically between 0.5 to 3 milligrams nightly, about an hour or two before bedtime. More assertive dosing regimens typically are discussed anywhere between 3 to 20 milligrams. Sometimes some authors will say even 5 to 20 milligrams nightly. And then in some individuals, they may go higher as tolerated. Usually higher doses are used in older individuals where their melatonin levels are naturally declining. I've seen protocols for individuals with chronic illnesses using between 100 to 300 milligrams of melatonin daily. It's typically not necessary for most people, but just understand that those do exist. And again, at least from my research, there is no LD50 for melatonin. So it's generally a very safe supplement to use. If you're working with kids, it's kind of like adults. The range is broad, but best initially dosed between, let's say, 0.5 to 10 milligrams. I think it is with anybody, particularly if you're working with people with chronic illness. You have to start low, go slow, and build up as tolerated. In some cases, levels in kids might go upwards of 20 milligrams. I mean, typically that's not needed, but again, there may be in certain circumstances. The 1 to 2 milligrams per kilogram dose is a range of consideration. Some authors will advocate for a broader range, 1 to 4 milligrams, for example. The biggest thing when it comes to melatonin, we know, is that in some people it can trigger vivid dreams. Some people will even get nightmares from it, for example. There's some discussion about people who are on SSRIs, so Prozac or Lexapro, for example, where melatonin might be contraindicated. Some of that data is probably loosely interpreted, because even in those people, they still can get benefit. And then, of course, you're always going to have patients that have sensitivities to any kind of supplement. So really what it comes down to is just understanding this information for what it does and understanding what melatonin does at the cellular level. And then you start using it clinically in your practice to see how people respond. From a supplemental standpoint, generally oral melatonin, if taken like a capsule, has fairly low absorption. I've read anywhere between 2.5 to 5% of it gets absorbed. That's different when you take it sublingually. So sublingual melatonin has a much higher absorption, and as well as liposomal. I've even seen some authors who advocate for suppository melatonin. So I think for most of us in practice, a sublingual tablet or a liposomal form of melatonin is going to be much better received, and the compliance in using it is going to be much higher. Some general recommendations that come about from some of the melatonin researchers, the feeling is that with increasing age, it's worthwhile increasing melatonin dosing. So let's say for somebody who's 40 to 45 years of age, for example, certainly a strong consideration to start implementing melatonin just as a preventative against cellular defects or diminishing aspects of cellular function. Some of the researchers will listen to, people who've been involved in melatonin research take melatonin as a daily product, and some research will even mention it. Usually older individuals, between 50 to 100 milligrams being self-dosed. So I think there's always more to learn when it comes to some of these supplements. Certainly that is the case of melatonin. I'm not advocating a specific dose for a given patient here or a specific disease, and not necessarily as a standalone. But I think the important thing from an integrative and functional medicine standpoint is that we as practitioners should really have a deep understanding about how things work, why they work as much as possible, so that we're better educated and we're better armed with things to do to try to normalize some of the biochemistry and physiology. Because things can become so complicated that sometimes we have a lot of either overlapping effects or we're not getting good clinical effects. And there's so much more out there in the natural world, supplement world, of things that can be used that are worthwhile. We just have to understand the mechanisms of why they're so helpful. And I hope this discussion, at least brief, on mitochondrial melatonin was interesting and may help you in practice moving forward with different types of complex patients. So a few of the resources that were used for this particular lecture, that one article I would strongly recommend, that Melatonin and Mitochondria, Mitigating Clear and Present Danger, that came from the journal Physiology 2020. That was Dr. Reiter. That first one is excellent. These other articles are excellent as well, and there's other research articles that I referenced within this lecture as well. Again, for those of you who are interested in our Mitochondria Mastery Course, this is starting later this month, in January 2023. A very in-depth course. Some of this information I took, part of this lecture came from that course or comes from that course. So go to mitochondriamasterycourse.com for more information. There's actually an ebook that you can download as well. There's a course curriculum and a lot of other information coming up about the Mitochondria Mastery Course. And then for our other courses or just general questions about our academy, you can email us at integrativemedicineacademy@gmail.com. And then the main website for our academy is integrativemedicineacademy.com. Well, thanks so much, everybody, for your attention. Again, I'm Dr. Kurt Woeller for Integrative Medicine Academy. Take care.